Hello, and welcome to this webinar on how genetics is helping us to understand Parkinson's disease. My name is Patrick Lewis, and I am a Parkinson's UK Research Fellow and academic based at the University of Reading in the School of Pharmacy. Today, I'm going to talk to you about some of the advances in human genetics over the last 2 decades that have really helped us to understand what causes Parkinson's disease, give you examples from my own research as to how this, in turn, is helping us to develop drugs for Parkinson's. Parkinson's disease is a neurodegenerative movement disorder that affects over 100,000 people in the United Kingdom. As with many neurodegenerative diseases, the chances of developing it increase as you get older. We've known about Parkinson's disease for a long time. Indeed, it is almost 200 years since James Parkinson first described the disease that now bears his name, in the paper shown on the left. The photo on the right is a Parkinson's patient that was seen by the neurologist Jean-Martin Charcot in Paris around 1880. Since this time, we have learned a lot about Parkinson's, both in terms of the symptoms, which include tremor or shaking, slowed movement, and problems with starting to move, as well as a range of psychiatric symptoms like depression, and the pathology of the disease, the changes in the brain that cause the symptoms. At its heart, Parkinson's is a fairly simple disease. It is neurodegenerative, which means that brain cells, neurons, die. This image highlights a region of the brain that is most affected by Parkinson's, a region called the substantia nigra. This sits right in the middle of the brain and is important for the control of movement. The image shown top right shows what the substantia nigra looks like in a healthy brain, with a characteristic dark color. Substantia nigra means black substance. This is due to the neurons in this area containing the chemical melanin, the same chemical that is found in skin. The image shown below shows what the substantia nigra looks like in the brains of people with Parkinson's. Almost all of this coloring has gone. This is because the cells containing that melanin have died. The most important impact of this is that the signals sent out by the substantia nigra using a neurotransmitter called dopamine don't happen, which in turn causes the movement problems that are the main symptoms of Parkinson's. We have known about this for decades, since the 1960s, and most of the drugs that can be used to reduce the symptoms of Parkinson's are based around replacing the lost dopamine neurotransmitter, for example L-DOPA, the most common drug for Parkinson's. Where we have made less progress is understanding why these brain cells die, and this is where genetics has helped. One of the most important tools with this regard is the humble family tree, an example of which is shown here. Hopefully you recognise this as the British royal family tree, and this is actually a good example of how you can use genetics to understand humans. Take, for example, Prince Philip here at the top, then look at Prince Charles, and finally Prince William. All of them share facial characteristics. They look similar. Most obviously, they all suffer from male pattern baldness. Part of what underlies this similarity is down to genetics. All three share a certain percentage of their DNA. DNA, or deoxyribose nucleic acid, is found in every cell in your body and provides the instruction manual for how each cell functions, from cells in your immune system that help protect against bacterial infection to the cells in your brain that control your movement. This instruction manual is made up of a chemical code represented by the letters A, T, G, and C. The order of these letters encoding the information that allows cells to function. As you can see, there is a lot of information. Over 3 billion of these letters make up the human genome. This information is organized as genes, bits of DNA that contain the instructions to make a particular protein, the machines and structures that give cells their shape and allow them to function. We think that there are around 30,000 genes in the human genome, each of which is coded for by a bit of DNA on a particular chromosome. the 23 long chains that the human genome is divided into. To make things a little more complicated, we actually have 2 copies of each chromosome in every cell, and therefore 2 copies of almost every gene. Now, the important thing to note about this code is that if you change it, you can change the function of the gene. These changes, called polymorphisms or mutation, depending on their context, are what provide the basis for much of the variety in humankind that you can see around you, and are passed from parent to child, with each child getting half of their genome from their mum and half from their dad. Here is a simple example of this, showing 2 different forms of genes responsible for eye colour being passed from generation to generation. The particular form a gene can take, here either a brown uppercase B or a blue lowercase b, can, in a simplified example like this, be described as being dominant, needing only 1 copy to produce a particular outcome or trait, or recessive, needing both copies of the gene to be the same for a trait to occur. In this example, having 1 copy of the big brown version of the gene will result in you having brown eyes, whereas both copies of the gene, remembering that we have 2 copies of almost every gene, have to be a lowercase b to have blue eyes. This works for simple traits. Geneticists call them Mendelian, after the Polish monk who first described this pattern of inheritance, like eye colour and, with a little bit of tweaking, things like hair colour or blood type. Importantly, this approach can also be applied to some types of disease. So instead of following eye colour through several generations, you can follow a particular set of symptoms passed from parent to child. For some diseases, like Huntington's disease, this is in a dominant fashion, where you need only 1 copy of the mutant DNA to develop the disease. For others, like sickle cell anemia, you need 2 copies of the mutated gene to develop the disease. For Parkinson's, the majority of patients don't have a clear family history of the disease, and so genetics is of limited use in understanding the disorder, although powerful new approaches are beginning to help us with this, of which more a little later. There are, however, a minority of patients, probably somewhere between 5 and 10%, who do have a family history of Parkinson's. This is an example of one of these families, from Lincolnshire in the United Kingdom. This is the most recent generation, here. This is the parents' generation, grandparents, and finally great-grandparents up here. Each of the diamonds in this family tree represents 1 member of the family. The diamonds which have been filled in are people who develop Parkinson's. As you can see, about half of the people in this family develop the disease, a clear indication to a geneticist that there is likely to be a genetic defect passed down through the generations. For this family, researchers at the University College London Institute of Neurology were able to take DNA samples and search through the human genome to identify the change in the code that was causing the disease, eventually finding a change on chromosome 12. This image here shows the actual change in the DNA that causes the people in this family who develop Parkinson's to develop the disease. The mutation is in a gene called leucine rich repeat kinase 2, or LRRK2, a cartoon of which is shown here. And this happens to be the gene that most of the research in my laboratory focuses on, of which more later. Neurologists and geneticists working in tandem have now identified a long list of mutations in genes throughout the human genome that are linked to Parkinson's, some of which are shown here in this table. These mutations explain some, but not all, of the 5 to 10% of Parkinson's cases that have a family history of the disease. But are these of any relevance to the 90% of people with Parkinson's in the UK who don't have a family history and are unlikely to have a genetic form of the disease? 5 years ago, I would have said, hopefully. Now I can say probably, at least for some of these genes. This is because of important advances in human genetics, such as the complete sequencing of the human genome, that have allowed us to move beyond simple Mendelian genetics, thinking back to the example of eye colour, and start to understand the much more complex influence of subtle changes in our genomes on the relative risk of developing diseases like Parkinson's. The idea is this, that there are powerful mutations, ones that change the way the brain functions, so much that having one makes it almost certain that you will develop Parkinson's. But there are also smaller, less powerful changes in our DNA that might increase the risk of developing Parkinson's over the whole of your lifetime by only a fraction of a percent. The way that geneticists identify these is by using something called a genome-wide association study, the basic principle of which is shown here. You start by taking DNA samples from lots of people with a particular trait, in this case Parkinson's, but actually the approach is applicable to pretty much anything that you can think of. You then take DNA samples from a group of people who don't have that particular trait, the control population. After purifying the DNA and using microchips, and based upon information from the Human Genome Project, you can then look at all of the genes in the human genome and identify small changes that are more common with people who have this particular trait than those who don't. Using some pretty fancy statistics, you can then calculate how important that risk is. What this produces is something called a Manhattan plot, shown here on the bottom. So called because it is meant to look a little bit like the skyline of Manhattan. This presents each of the human chromosomes numbered from 1 through to 22, with the red skyscrapers representing bits of these chromosomes that are associated with an increased risk of Parkinson's. In some cases, we can identify particular genes that are linked to these skyscrapers, and this is where it gets interesting. Quite a few of these skyscrapers are located in genes that we already knew about from the familial forms of Parkinson's that I was talking about earlier. This, admittedly slightly smaller skyscraper on chromosome 12 represents the gene LRRK2. Why is all this important? What it does is it provides confidence that if we can understand what some of the rare mutations in families who have Parkinson's do to the brain, and can find a way to stop or reverse what it is the mutations are doing, then we might be able to use the same approach in the 90% of people who don't have mutations. Put another way, you have mutations up here in red that can cause Parkinson's. These are the ones that are identified in families who have a genetic form of the disease. You then have risk factors for Parkinson's, shown down here in green. These are identified using genome-wide association studies. The fact that we are finding some of the same genes in both groups means that there are changes in how brain cells function that are similar in both, which brings us back to LRRK2, which is shown here and here. We've learned a lot about LRRK2 since 2004, when the mutation that causes this family's Parkinson's was identified, and we first found out how this gene was linked to the disease. We now know that mutations in LRRK2 are the most common genetic cause of Parkinson's. We also know that LRRK2 is involved in the risk of developing Parkinson's across your lifetime, even if you don't have a family history. Coupled to this, LRRK2 has aroused the interest of pharmaceutical companies, as it has two pockets called active sites that can be targeted using drugs. This allows scientists to turn LRRK2 on and off, which might be important in a clinical setting. A lot of research remains to be done on this, so it is too early to tell at the moment. However, these are very powerful tools for understanding what LRRK2 does. LRRK2 is distinctive as a Parkinson's gene for another reason, in that there is a very public face for mutations. This is Sergey Brin, one of the co-founders of Google. A few years ago, his mum developed Parkinson's disease. Sergey Brin is Ashkenazi Jewish by descent, and, for a number of reasons, Ashkenazi Jews are more likely to have mutations in LRRK2 than the general population. Sergey Brin's wife happened to be a geneticist, and she suggested that he should have his LRRK2 gene analysed, revealing a mutation in LRRK2. This mutation, affecting one of those binding pockets I was talking about in the previous slide, means that by the time he is 80, Sergey Brin has an 80% chance of developing Parkinson's. This highlights a number of things. First of all, technology has advanced to the point where this kind of genetic analysis will become commonplace. The company established by Sergey Brin's wife, 23andMe, already offers genetic testing of the LRRK2 gene. Second, it raises a number of ethical issues surrounding the genetic diagnosis of diseases that we don't yet have a curative treatment for. This is a huge area of debate at the moment, and one that is beyond the scope of this talk, but it is certainly an area of medical ethics that deserves a lot of attention. There are lots of scientists working on different aspects of Parkinson's, but I'll give you an example of how my lab is trying to understand the causes of the disease using genetics. And for this, we return to LRRK2. My research aims to try and understand what this gene does in human cells, and why mutations in it are so damaging. My group does this in a variety of ways, one of which is looking at human brain cells, shown up here on the left, to see what happens when you switch LRRK2 off using drugs, or when there are mutations in it. We can grow these cells in the laboratory, in dishes, shown here in the capable hands of Joe Giavelli, a former student in the lab, allowing us to look inside these cells to see what LRRK2 is doing. This is a drawing of your common or garden neuron. In fact, there are lots of different types of neuron, by some estimates over 10,000, but this shows most of the bits that all neurons share. It is made up of a cell body, shown here, extensions called dendrites, which are these bits, which act to collect information from other cells, and a long extension called an axon, which the neuron uses to send information to other cells. To give you an idea of scale, the cell body of the neuron, shown here, varies in size from about 4 microns, a micron is 1/1000th of a millimeter, to about 100 microns, so 0.1 millimeter in size. Basically, very, very small. When you zoom in on the cell body, you can see there is a lot going on, some of which is shown here. There is DNA, coiled up and stored in the nucleus, shown here in blue. The endoplasmic reticulum, which is an area where the DNA code is turned into proteins. There are also mobile power stations, called mitochondria, which generate the energy for the cell. Although I am not going to talk much about these are very important for Parkinson's disease. There is also a waste disposal system, made up of membranous bags called autophagosomes and lysosomes. Autophagosomes act a bit like rubbish trucks. This is an example from Maidenhead, where I live. They collect up broken down proteins and bits of the cell that aren't working properly, and take them for disposal and recycling, carried out by the lysosomes. The way in which they do this is by forming an open bag, shown here, made up of a membrane from inside the cell. One of the researchers in my lab, Claudia, thinks this looks a lot like Pac-Man, and she has a point. And in a way, it is a good analogy, as the autophagosome goes around the cell looking for rubbish to eat up. For example, these are 2 mitochondria. If these mitochondria go wrong, autophagosomes are one of the ways in which the cell can collect these up. The bag is then closed, and the rubbish taken to the lysosomes, where a type of protein called a protease, amongst others, help break down the waste, allowing it to be recycled. What does this have to do with Parkinson's disease and LRRK2? What I've shown here is a photo of some of the changes that happen in the brains of people with Parkinson's. These brown blobs in the brains of people with Parkinson's are called Lewy bodies. They are made up of sticky lumps of a protein called alpha-synuclein, shown as a cartoon over here. And we think that these sticky lumps are one of the reasons why brain cells die in people who develop Parkinson's. Autophagosomes are one of the ways in which these sticky lumps can be gathered up and got rid of. Similar to the mitochondria I showed you a couple of slides ago, the lumps are enclosed and then disposed of. Now, if the autophagosomes aren't working for some reason, then the sticky lumps of alpha-synuclein can accumulate, which is likely to be bad for the brain. And this is where LRRK2 comes in. Work in my lab, funded by Parkinson's UK and the Michael J. Fox Foundation in the US, as well as experiments from lots of other labs around the world, suggests that one of the jobs that LRRK2 does is to help control how autophagosomes function, a bit like the council office working to send out the rubbish trucks on the right day and to the right place. What the mutations in LRRK2 might be doing is disrupting this control system, causing cells to accumulate damaged proteins and, in some cases, alpha-synuclein. As I mentioned before, pharmaceutical companies and academic labs have developed drugs that can target LRRK2, switching it off. The one on the left here, called LRRK2/1, was developed at Harvard University, the one on the right by GlaxoSmithKline. These drugs are important for understanding what LRRK2 does. We use them in our experiments examining autophagy, published in these papers, but also have potential as an experimental treatment for Parkinson's. At the moment, we don't know if switching LRRK2 off will help slow down the death of brain cells in Parkinson's, and there is a lot of research going into this area at the moment to try and establish whether this is the case. And it is important to remember that LRRK2 is just one of the many drug targets that are being investigated for Parkinson's. But it provides a good example of how genetics is helping us to understand Parkinson's and helping us to get further down the road towards a treatment. With that, I'll finish up. I'd just like to thank my lab, in particular Claudio Manzoni, shown here, and collaborators, in particular Rina Bandapadai at the Institute of Neurology, over here on the left. And of course, all of the charities and funding bodies who have made this work possible. If you have any questions, feel free to drop me an email at p.a.lewis@reading.ac.uk or visit my website, where copies of most of the papers that my group has published are available to download. Thanks very much for your attention.